Hello friends, today we are going to see molecular tool known as PCR. PCR is nothing but an polymerase chain reaction. This molecular tool is used for amplifying the DNA fragments of our interest. Say like if any scientist want to make an experiment where he has a very small amount of DNA sample, but for the experimental purposes he needs a large amount, so he can carry out PCR process to amplify that DNA sample. This process was invented by Karen Mullis, scientist, in the year 1983. So we will see what are the requirements for this process. This process requires a target DNA, some primers and an polymerase enzyme. Along with this polymerase enzyme, they require certain nucleotides. We can call them as DNTPs. For this purpose, initially we required an target DNA which needs to get amplified. Then there are primers, we can say them as forward and reverse primers. These primers are nothing but 10 to 20 nucleotide sequences which will be complementary to the templates. Taq polymerase here is an enzyme which is derived from an organism Thermus aquaticus, which is thermostable. This is very specific for this reaction because this organism can survive high temperature, or we can call it as a thermostable organism. So that's where Taq polymerase is being used. So moving towards the process, this PCR process is differentiated into three main phases. First one is denaturation, second one is annealing, or you can call it as a renaturation process, and the third one is synthesis, or it is also called as an extension phase. So starting with the first process, denaturation. Denaturation is nothing but a process where double-stranded DNA is allowed to undergo a very high temperature, and due to that high temperature it start getting separated, means the two strands or double-stranded DNA, the bonds between that nucleotides will get melt and it will get separated out, and you get a single-stranded DNA. The temperature here is 96 degrees Celsius. After getting separated, where you get single strands, these templates can be allowed to get annealed with the primers. Primers are nothing but 10 to 20 nucleotide sequences which will be complementary to these types of templates. The primer annealing process is carried out at 55 to 65 degrees Celsius, where, or we can call this process as a renaturation process, where both the template strands will be allowed to get attached with the primers. These are the primers. Suppose one would be forward and one would be the reverse primers. These both primers will move in anti-parallel direction. As we know, DNA polymerase enzyme always works in certain direction, like 3' to 5' or 5 to 3', where the synthesis of the strand here it is done in 5 to 3' direction. So this primer will get annealed with the template, where both the strands would be complementary to each other. After annealing process, there would be an synthesis process, or we can call them as an extension process. After the primers, when it will get attached with the templates, with the help of Taq polymerase enzyme, these primers would extend the strand, and after one cycle you get double-stranded DNA. Means from this double-stranded DNA, from 2 copies we get 4 copies, 2 and 2. So after one cycle, as we get 4 copies, so around 25 to 35 cycles are being carried out, and this whole process requires around 2 to 3 hours. Say like after this double-stranded DNA, after one cycle you get 2 types of double-stranded DNA from using this as an template. So like this 25 to 35 cycles are being carried out for one PCR whole process, and you can get billions of DNA fragments of your desired interest. So after receiving all these samples can be analyzed with the process of gel electrophoresis. As we know, gel electrophoresis is an tool where you can analyze all types of DNA fragments to check whether the samples are properly isolated or not. So thank you.